How do we really measure success, when we talk about these cutting-edge cancer treatments?
Right. Is it just about shrinking the tumor?
Exactly.
Mm.
Or is there a bigger picture, something more complex, especially when you look long-term?
Mm-hmm.
Well, today we're taking a deep dive right into that question.
Okay.
Our mission is to unpack the final long-term results from the RELAY study. It's a major global clinical trial.
Focused on advanced non-small cell lung cancer, right? Specifically the EGFR mutated kind.
That's the one. We want to pull out the most important insights from all this complex research, especially around how long patients actually live.
And what factors influence that?
Yeah, because it turns out it's not always straightforward.
And for this deep dive, we're sticking entirely to one key source. It's the article titled Relay, Final Overall Survival for Erlotinib plus Ramucirumab or Placebo in Untreated EGFR Mutated Metastatic NSCLC.
So this is the final word on the long-term follow-up.
It is. It gives us that crucial overall survival data.
Okay, let's set the stage then. Non-small cell lung cancer, NSCLC. It's the most common lung cancer type.
Right. And for patients with those specific EGFR mutations, these targeted therapies have been, well, game changers.
Like an on switch for cancer growth, basically.
Yeah, that's a good way to put it. And EGFR TKIs, the tyrosine kinase inhibitors, they're designed to flip that switch off.
So the RLA study looked at a combination, two different drugs.
Exactly. Erlotinib, which is a first-gen EGFR TKI, and Ramucirumab.
Okay.
That one's an antibody targeting a different pathway, the VEGF pathway.
VEGF, right. So the idea was a kind of dual blockade, hitting two targets.
Precisely. Block both EGFR and VEGF pathways, aiming for a stronger anti-cancer effect, tackling growth and the blood supply.
And the trial itself, NCT 024114448. Pretty robust design.
Yeah.
Global, double-blind, placebo-controlled.
Phase 3, yeah. Very rigorous. They enrolled 449 patients, split them 1-to-1.
That kind of design is crucial for reliable results, isn't it?
Absolutely. It minimizes bias as much as possible.
Okay.
But there's one specific detail in the design that's quite important to remember.
What's that?
Patients with CNS metastases, cancer spread to the brain, they were excluded.
Ah, right. That is significant for this patient group. How might excluding them affect how we look at the overall survival results later on?
Well, it could definitely have an impact. Brain metastases often mean a poorer prognosis. They affect survival.
Okay.
So by excluding them, the patients in RLA might have started out, on average, with a slightly better prognosis than you'd see in, say, a real-world population of all EGFR mutated patients.
Got it. Something to keep in mind. Now, before we get to the main event, the overall survival, let's just remind ourselves, RLA already had a big win, right?
It did, yeah. Its primary endpoint was progression-free survival, PFS.
Which is how long patients live without the cancer getting worse.
Exactly. And the combination therapy, Ramucirumab plus Erlotinib, it hit that endpoint hard.
How much better was it?
Median PFS was 19.4 months for the combo arm, compared to 12.4 months for placebo plus Erlotinib.
Wow, okay. That's a 7-month difference.
A big difference. Statistically significant, clinically meaningful. That's what led to regulatory approvals worldwide.
Okay, let's unpack this then. So this combination therapy already showed a clear benefit in delaying cancer progression. But the real long-term question, what patients really want to know, is about actually living longer, right? Overall survival.
Exactly. And that's the core focus of this final report. Overall survival, or OS. It was a key secondary endpoint.
And the data's mature now.
Yes, very much so. They followed patients for a median of over 45 months. By the cutoff, 297 deaths had occurred, which is about 66% of the patients.
So what did the final OS numbers show?
Okay, so for the Ramucirumab plus Erlotinib arm, the median OS was 51.1 months.
61.1, okay. And for the placebo plus Erlotinib arm?
It was 46.0 months.
Okay, so there's a numerical difference there. Well, 5 months longer median survival with the combination.
There is a numerical difference, yes. And yet, and this is the critical part, that improvement in overall survival was not statistically significant. The p-value was 0.864.
Not significant. That's a really important distinction, isn't it?
It absolutely is.
Can you help us understand why that matters so much?
Yeah.
Especially when, like you said, those median survival times in both arms, 51 months, 46 months, they seem quite long.
They are quite long, actually. Longer than reported in some other similar trials.
So why wasn't the difference significant? What's going on there?
Well, that's the fascinating question the report tackles. First off, a crucial point. The RRelay study just wasn't designed, wasn't powered to detect a statistically significant OS difference.
Okay, so its main goal was PFS.
Right. But even beyond the study design, the really big factor seems to be what happened after patients stopped the study drugs.
You mean the treatments they got later?
Exactly. There was a surprisingly high rate of subsequent therapies. Over 80% of patients in both arms got some kind of treatment after discontinuing the study medication.
80%. Wow.
And many got multiple lines of treatment. The median was 2 to 3 different lines of therapy afterwards.
Okay.
And here's the kicker. Over half the patients in both arms eventually received osimertinib.
Osimertinib, the third-generation TKI, which became the preferred first-line standard after RLA had already started.
Precisely. So you have this really effective drug becoming available and widely used during the trial's follow-up period. It changed the whole landscape.
They must have had a huge impact on the results.
A massive impact. Look at the numbers. For patients who did get osimertinib later on, their overall survival was really long. 57.3 months if they started on the Ramucirumab combo, and even longer, 61.3 months if they started on placebo plus Erlotinib.
Oh, 61 months for the placebo arm group that got osimertinib later.
Right. But now look at the patients who didn't get osimertinib later.
Okay.
Their OS was much shorter, only 36.7 months for the Ramucirumab combo group and 30.3 months for the placebo group.
That's a huge difference. So if I'm getting this right, it's like the effectiveness of those later therapies, especially osimertinib, just washed out or maybe masked any survival difference from the initial study treatment.
That's exactly what the authors suggest. The powerful effect of subsequent therapies, particularly osimertinib, likely obscured or confounded any potential OS benefit from the initial RAM plus BRL combination.
It basically leveled the playing field long term.
It seems so. This is a major challenge in oncology trials now, you know? Treatments are evolving so fast.
It sounds like trying to prove an overall survival benefit in a field moving this quickly is like trying to hit a moving target.
It really is. When new, super effective drugs become standard care during your trial's follow-up, they can lift survival outcomes for everyone, making it incredibly hard to isolate the true long-term impact of the drug you were initially testing.
Even if that initial drug clearly helped with PFS, like we saw here.
Exactly. The PFS benefit was clear, but the OS gets muddied by subsequent treatments.
That's a really critical point for interpreting trial data today. Okay, so even though there wasn't an overall statistically significant OS difference, the researchers did dig deeper, right? They looked at subgroups.
They did, yes. Trying to see if maybe certain groups of patients responded differently.
Like based on their specific EGFR mutation?
Correct. They looked at the two most common types, the L858R mutation and the exon 19 deletion.
And what did they find there? Any hints?
Well, for patients with the L858R mutation, there was a trend, a directional improvement in OS for the Ramucirumab plus Erlotinib arm.
How much of an improvement?
Median OS was 51.6 months versus 45.8 months for placebo. The hazard ratio was 0.87.
Okay, suggesting a potential benefit. But for exon 19 deletion?
No OS benefit observed there. In fact, the median OS was slightly lower numerically in the combo arm, 49 months versus 51.4 months. Hazard ratio was 1.13.
Interesting.
Now, these weren't statistically significant findings. The confidence intervals overlapped one, but it does kind of hint that maybe these mutation subtypes respond differently to this specific combination.
It really highlights that not all EGFR mutations are the same. Fascinating. And then there was this other factor, the TP53 co-mutation. That adds another layer, doesn't it?
It certainly does. TP53 is a really important gene.
What does having that mutation tell us about a patient's outlook?
Well, TP53 mutations were found in nearly 43% of the patients in our relay. And generally, having that co-mutation meant poorer overall survival outcomes, regardless of which treatment arm they were in.
How much poorer?
Median OS was about 42 months for those with the TP53 co-mutation, compared to over 60 months for those without it. So it definitely marks a more aggressive disease, a poorer prognosis.
Right. And did the combination therapy show any particular effect in this poorer prognosis group?
Yes, interestingly it did. Similar to the L858R group, there was a directional improvement in OS for Ramucirumab plus Erlotinib, specifically in patients with the TP53 co-mutation. The hazard ratio was 0.83.
But not in the patients without a TP53 mutation.
Correct. No such trend was seen in the wild-type TP53 patients.
So if we try to connect the dots here, does this suggest the benefit of adding Ramucirumab might be more pronounced in these higher-risk patients, maybe those with L858R or TP53 mutations?
That's a very plausible interpretation, I think, and there might be a biological reason for it, especially with TP53.
Okay.
Well, TP53 mutations are known to be involved in angiogenesis. That's the process of forming new blood vessels that feed tumors.
Okay.
And these mutations can actually increase the activity of the VEGF pathway.
Which is exactly what Ramucirumab targets.
Precisely. So in patients where that VEGF pathway is already potentially upregulated due to a TP53 mutation, adding a VEGF inhibitor like Ramucirumab might provide a greater benefit.
That makes a lot of sense biologically. It's really insightful. These subgroup analyses, even if they're not definitive proof on their own, they really point towards a future of more personalized treatment, right? Looking beyond just the main EGFR mutation.
Absolutely. It underscores the need to understand the patient's full molecular profile.
Okay, let's shift to practicalities.
Yeah.
We saw the PFS benefit. What about the downside? The safety profile of adding Ramucirumab.
Yeah, that's important. The overall safety was generally consistent with what we know about Ramucirumab and Erlotinib separately.
But was it worse with the combination?
It was. There were higher rates of grade 3 or higher adverse events, with the combination 76% versus about 56% in the placebo arm.
76%. What were the main issues?
The key differences, the things seen more often with the combination at grade 3 or higher, were hypertension, that was about 24%,
Right.
diarrhea at 7.2%, and that acne-like skin rash, dermatitis acneiform, at 16.3%.
Any other serious concerns?
The report does mention one additional fatal adverse event, a case of sudden death in the Ramucirumab arm since the primary analysis was done.
That's serious. So a definite increase in toxicity. But what about the patient's experience? Did this impact their quality of life?
That's the crucial counterpoint. Despite the higher rates of these side effects, the patient-reported quality of life scores were not statistically different between the two arms.
Really? So even with more side effects, patients didn't report feeling significantly worse overall.
Apparently not, based on the standardized questionnaires they used. It suggests the side effects, while more frequent, were perhaps manageable or didn't drastically impact their overall well-being compared to the control group.
That's important context.
We should also mention the practical side. Ramucirumab is given intravenously, which means regular clinic visits. That's definitely an inconvenience, as the paper puts it, compared to just taking pills.
A very real factor for patients, yeah. Okay, thinking about treatment sequencing, where does this relay regimen, RAM plus RRL, fit in today's world, especially with osimertinib being so dominant up front now?
Well, one potential advantage of using the RLA regimen first is that it keeps osimertinib in reserve for later.
Ah, so you save the big gun.
Potentially, yes. It preserves osimertinib and also standard chemotherapy options for subsequent lines of therapy. It offers flexibility in sequencing.
Which could be valuable for extending overall benefit across multiple lines.
It could be. But as we've discussed, the landscape has shifted dramatically since RLA started back in 2016.
Right. Osimertinib is the first-line standard for many now.
Absolutely. So while Relay gives us valuable data, especially these subgroup hints, it's being interpreted against this constantly moving backdrop.
Is there ongoing research looking at this combination in the current context?
Yes, definitely. There's a study called REVEL 858R, for example. It's directly comparing Ramucirumab plus Erlotinib against osimertinib, but specifically in patients with that L858R mutation.
Ah, targeting that subgroup where Riley hinted at a benefit.
Exactly. And there are other trials exploring different combinations, too. This field is incredibly active.
Okay, so let's try and wrap up this deep dive into the final RLA results. Key takeaway number 1. The combination of Ramucirumab and Erlotinib clearly provides a significant progression-free survival benefit. It delays the cancer growing.
Correct. That initial finding holds strong.
But, takeaway number 2, it did not show a statistically significant improvement in overall survival in the final analysis.
Right. And that seems largely because so many patients in both arms received highly effective later treatments, especially osimertinib.
Which essentially masked or diluted any potential OS benefit from the initial therapy?
That seems to be the most likely explanation, yes.
However, digging deeper into subgroups, the data suggests potential OS improvements, maybe directional benefits, for patients with specific characteristics.
Namely, those with the L858R mutation or those with a TP53 co-mutation, poorer prognosis groups, perhaps.
Exactly, which highlights this growing need to look beyond just the primary EGFR mutation.
It really drives home that treating EGFR-mutated lung cancer isn't about one single best drug, but about finding the optimal sequence of therapies for each individual patient.
It's so complex, isn't it? All these interconnected factors. Finding a good treatment is one thing, but finding the best order of treatments.
Especially with new options emerging all the time.
So thinking about all this,
Yeah.
the new drugs, the better understanding of LE588R, TP6580, how do we, as a medical community, actually take all this information and use it to truly personalize care? To really maximize long-term survival and move away from a one-size-fits-all approach is a huge challenge.
It is. Integrating genomic data, trial results, sequencing strategies.
Oh, yeah.
It's complex.
So for you listening, what stands out from this deep dive? What's the most impactful insight from Arle when you think about future treatment decisions in this space?